recombinant DNA and its applications, and in this session we'd like to look at the construction and usage of genetic libraries. So what a library actually is, I know you know what the regular one is, the big building, but it's a collection of clones, and they don't have to be, but we're going to say that those clones are individual plasmids, each of which carries a single portion of the genome of a particular organism. So in other words, the net sum of all of these plasmids together would represent all of the information in the genome of the organism. You can see the analogy of a library if you say that each of the books in a library represents sort of the same idea as one of the clones here, and that the whole bunch of books in the library represents all of the knowledge that exists, something like that anyway. So you can construct lots of types of libraries. The most inclusive one, and the kind we'll discuss first, is called a genomic library, meaning that somewhere in those books in the library, those clones, you will find every single gene and other DNA sequence present in the genome. So I think I just said this, but all of the clones taken together contain at least one copy of every DNA sequence in the genome. But you might say, so what? What's the difference between having that and the genome? Well, we're about to find out. You construct the library using the same principles as we used in cloning of specific genes. So we can just remember those tools and things that we used. So how would you do this? And then we can discuss why you might want to do it. The first thing you'd do is isolate total genomic DNA from an organism. Let's just say it was a human to go along with what we've been doing. Now in this case, you'd want to digest that genomic DNA with a restriction enzyme. It doesn't particularly matter which one in this case, but what you can imagine that would do is create literally thousands, if not more, fragments depending on the size of the genome, the frequency of a particular restriction enzyme recognition site. In fact, you can use those variables if you want to have more or less fragments. And then, so you've got all these different fragments. Incubate the fragments with appropriate cloning vectors in the presence of ligase. So it's almost as if we're doing what we did last time when we cloned a gene, just on a large scale. So the bottom line is you're just taking all the fragments, potentially hoping that they all get in to a single, each of them gets into a single cloning vector, meaning you might have thousands of plasmids, but each of them took up one of the pieces that we created when we restriction digested the entire genome. And as we saw before, then you would use the transformation process to introduce these many recombinant plasmids into bacterial hosts. So what the result would be is a collection of clones, but really what it would be is a collection of clones inside of bacterial cells representing every single sequence in the genome. So in other words, one cell may have taken up a recombinant plasmid that includes one particular gene. We'll have another cell that's taken up a different one, etc., etc., etc. So here's a great picture, I think, that explains, well, pictorially how to do this. So it doesn't have to be, but we're going to take a person here, human DNA, and you have total genomic DNA. You could get it from any place that it exists, which is every cell in your body pretty much. And we're going to create recombinant DNA molecules. Well, first thing we're going to do is chop it up, right? Using a restriction enzyme. And in this case, you can see that all the blue fragments that are over here represent, there will be a lot more of them, but they represent pieces that were made by chopping up the human genome with the restriction enzyme. Then we're going to take plasmid DNA, which is going to be our cloning vector, and we're going to digest that with the same restriction enzyme, right? So the red fragments over here represent the fact that actually what they represent is that these cloning vectors should have one site for the relevant restriction enzyme, right? So they're really, it's they're linear pieces there, but what we've really done is opened each of them up so that it's ready to accept one of the blue fragments, in this case, one of the pieces of the human genome. Okay, so now we're ready to look over here. We're doing basically, I hate to keep saying it, but what we did before, which is create recombinant DNA molecules, in other words, plasmids harboring the human sequences, by joining the fragments together with ligase, just like we did. And what we'll end up with is a population of plasmids now sort of randomly containing different pieces of the human genome. You could see here, now what you can see here is that, look, some of the plasmids did not in fact take up a piece. This one took up one piece, this one took up another, etc., etc., etc. Then we could take E. coli. I mean, you don't have to use E. coli, but everybody does since it's readily available and we know everything about it. Transform various cells with these recombinant plasmids. So you'll then have, and do the whole thing, transform, select, so you know which one's got the plasmid, and then you'll end up with a whole collection down here of bacterial cells, each of which harbors a plasmid, and each of those plasmids harbors one portion of the human genome. So I just wrote down here for fun, this is maybe this is where our human insulin gene that we were working with last time came from in the first place. This fragment happened to harbor the human insulin gene, so that ends up in one of our clones. The CFTR gene is the one that, when mutated, causes cystic fibrosis, so maybe that ends up in another place. And I wrote question marks, meaning we don't know. But what we do know is that every single gene and other sequence in the human genome is represented somewhere in that library. So I think that's understandable. What are you going to do about it? Do with it, I should say. Well, before we ask that, I mean, partially we're going to address these things in the applications session. But before we ask that, there are some other types of libraries that you should be aware of and understand the difference. So chromosomal libraries are pretty popular. And if you've already narrowed down a gene's location to a particular chromosome, and you can do that by more traditional mapping techniques often, or there are a variety of ways, what you can do is just get a library of sequences from that chromosome. If you know you're interested in a particular gene, in humans obviously there are 23 different chromosomes. Say you knew that your gene of interest was on chromosome 7. So if you get a library just of all the material in chromosome 7, you don't have to parse through all of the information on the other chromosomes. A very important and interesting type of library, and just a concept in general, is that of a cDNA library. cDNA stands for complementary DNA. So if you're looking for a protein coding gene, which you're not always, but you often are, you can save yourself a lot of time by using cDNAs. So protein coding genes are expressed, and if they're expressed, there must be a messenger RNA that's made, right, to make the protein. Now in humans this would be tissue specific, and you can use that as well. In other words, you could make a cDNA library from the liver, which would have all of the proteins. I should say what it would have is all of the DNA sequences corresponding to proteins that are expressed in the liver. But how you do it is really cool. You get the relevant messenger RNAs and then use the enzyme reverse transcriptase, which naturally occurs in retroviruses, to backwards transcribe and make that RNA into DNA, but it's cDNA. And look at the beauty of this. Not only are you gotten rid of all the extraneous genomic sequences, I say. In other words, we haven't discussed this a lot, but sometimes the term junk DNA is used. I don't like that, but let's just say that the majority of sequences in the human genome don't in fact encode genes that make proteins. They do other things, or maybe they don't, but there's a lot of material that is not relevant if you know that that's what you're looking for. But also another important point, and I don't know if we ever mentioned this, is that if you're going to put a DNA, it depends what you want to do with it, but let's say we're using human insulin. If you pop the real human insulin gene into a bacterium, as we implied last time, you're going to have a problem, and that is introns are present in that gene, and bacteria don't know how to remove introns. So I should have, well, we have to go one step at a time. But in reality, they have to make the cDNA for the human insulin gene if you want to industrially produce human insulin from a bacterium, because you've effectively removed the introns, right? If you have mature messenger RNA coding for insulin, it's already been processed. Now you're going to back transcribe it to DNA and the introns will be gone. So cDNA is very cool and has a lot of applications. Now, more to the point of what you do with it once you have it, you're going to screen a library. So what it allows you to do is identify, search for, and subsequently work with a gene of interest. So I'm into the insulin gene here, but here's an interesting example of what you might want to do. Say you want to identify and characterize the insulin gene in a horse. Now I just made that up, but you might be interested in that. But the point is, it could be anything. But let's just say we know about the human insulin gene, but we don't have a lot of information about the horse insulin gene. For example, where it's located in humans on whatever chromosome is not going to be the same as in a horse or in any other organism that makes insulin. So what would you do if you wanted to work on the, say you want to sequence the horse insulin gene and compare it with human? Well, get a horse genomic library, right? You have all of the sequences of the horse genome, a very tiny bit of which would be the horse insulin gene, and screen it. Now, what you would do is use the human insulin gene, which we have already characterized, right? And we assume, and it's a good assumption, that human insulin, given that it has the same function in horses as it does in humans, would be a gene that's not identical, but is going to be similar. So, basically, I'll show you the procedure, but you're going to use the human insulin gene to basically look around at the library and find something that looks like itself, and only one or a few of the clones in the library out of thousands would be identified. And you'd know that the horse gene is right in that one. You're separating it away from the whole rest of the genome, and then you could work with that clone, either further refine the clone or use that one to do what? Whatever you want to do, right? Say sequence the horse gene. So how do you literally screen the library? There are a variety of modern ways to do this without radiation, but using a radioactive probe, you're always using a probe, is a common thing to do. But what the probe is a single-stranded DNA molecule containing the human insulin sequence. It has to be single-stranded because what we're going to do eventually is make the horse DNA single-stranded as well, and that way, even though it's not a perfect match, the human gene will hybridize, cross-hybridize with the horse DNA because they're similar enough, certainly as compared with any other sequence in the horse genome. So that's what the probe is. And if it is radioactive, well, let's go through the steps. That's the idea. So you would plate out bacteria carrying the clones, all of them. You might have 10 different plates. I don't know. Then you transfer the colonies to membranes. Now the term membrane is a little strange here. What it really is a piece of paper. It's fancy paper, but you put the paper over where the bacteria are growing, and it actually transfers the cells along with their DNA onto this piece of paper because it's a lot easier to work with the paper. We need to, in the process, lyse the cells so the DNA comes out, and also denature the DNA because of what I said before. We want it to be single-stranded so that we can probe it, and then mix it together with the radioactive probe and each of the clones, right? So, assuming this was radioactive, it's only going to bind to the places where the relatively complementary horse insulin gene exists. That's not going to be most of the clones, right? So only the clones will light up, meaning what you really do is you put a piece of X-ray film here. If there's radiation there, it will expose the film and you'll be able to see which ones of your original colonies have bound to the human insulin probe, and therefore you'll know that those are the colonies harboring the horse version. So autoradiography basically means it's auto just because you don't really have to do anything except put a piece of film there and the radiation exposes it. So only complementary sequences will bind the probe. The radiation will tell you where that's occurred, and that'll tell you which clones you can now focus on and just throw all the rest away. So here's a great picture, I think, sort of pictorially illustrating this. We have a plate containing the clones here. Now, this is one plate. There probably would be a bunch of them that you would have to do this to. And I'm leaving out a couple, well, not leaving them out, but we're sort of streamlining the process a bit here. So transfer the clones to the membrane, right? Don't forget they would have to be lysed and denatured as well. Add the probe solution. So here's the radioactive probe, right, washing around on this. And it's only going to bind to one or two places where the horse insulin gene exists. So in this case, drew them in blue, there were two clones that, quote, lit up. Now, why would there be two? Well, sometimes there's one, sometimes there's two. It's possible there even could be more. The reason is because when you initially made the library, you sort of randomly chopped this up into pieces, not really knowing, what restriction sites were where. So if you happen to, and it might happen and it might not, if you happen to cut in the middle of the insulin gene, say half of it might have ended up in one clone and half of it might have ended up in the other clone. And that seems to be the case of what happened here. But that's fine. So you just take these two out of the hundreds and hundreds that you have, and you know that those are the ones of interest. You grab those. Now, it's on the piece of paper, so what you really have to do is go back, figure out which one it was on the plate, and then take those cells. Well, actually, we don't need to worry about the technicalities because what I say here is infect bacterial host with clones containing the gene of interest. So you could do it that way as well. You could isolate this DNA, re-put them, if that's a word, back into some new bacteria, or you could go back and find the original clone, all right? But the bottom line is you're going to end up with bacteria harboring the plasmid that is the clone of interest, meaning all or part of the horse insulin gene would be on that clone. So you screened the library. In the application section, perhaps obviously there are a lot of things that you could do with any of these techniques. So, for example, I sort of alluded to this, but I just want to say it. If you watched the last one about how to clone the gene, we said we're going to clone the human insulin gene. But we never really addressed, we said, yeah, we cut it out of the genome. But in preparation for doing that, probably one thing you could have done was to make a human genomic library and sort of use some sequence information, or maybe do the reverse of what we did. If we know about the pig gene more than the human gene, we could use that to sort of find where the human gene is. And that would be our initial sort of pre-clone that we would then do some further characterization of. So that's how to make and screen a genetic library. If you're going in order, we're going to talk next about how to analyze DNA at the molecular level, and I look forward to seeing you then.